Myself, Ananya Joseph, first MSc Biology, St. Mary's College, Thrissur. Today, we're discussing the topic History and Scope of Microbiology, Microscope, and the Microscopy, the principles and application for the study of microorganisms. So, first, what is microbiology? Microbiology is the study of living organisms of microscopic size, it including the bacteria, fungi, protozoans, algae, etc. Microbiology includes the distribution of microorganisms in nature, their relationship to each other and to the other organisms. And the microbiology is closely associated with the health and welfare of the human beings. Some microorganisms are beneficial and some are detrimental. Next is the scope of microbiology. Microorganisms affect the well-being of people in a great many ways. The diversity of their activities ranges from causing disease in human, other animals, and plants to the production and deposition of minerals, the formation of coals, and enhancement of the soil fertility. Next is the major field of microbiology, that is medical microbiology, aquatic microbiology, aeromicrobiology, food microbiology, agricultural microbiology, industrial microbiology, and the exomicrobiology. The medical microbiology consisting in the study of causative agents of disease, diagnostic procedures, and etc. And the aquatic microbiology including the water purifications, microbiological examination, biological degradation of waste and ecology. Then the aeromicrobiology including the contamination and spoilage, dissemination of disease, etc. The food microbiology including the food preservation and preparation, the foodborne diseases and their prevention, etc. Agricultural microbiology means soil fertility, plant and animal diseases, etc. The industrial microbiology means the production of antibiotics, vaccines, fermented beverages, production of protein and hormones by genetically engineered microorganisms. And the last is the exomicrobiology, that means the exploration for life in outer space, etc. This is an chronological order of history of the microbiology. Uh, from the 1857 to 1911, this timeline of is known as the golden age of microbiology. Next, we see the microscopes and microscopy. Microscope is an instrument most characteristics of the microbiology laboratory. And what is the magnification? Magnification means enable us to see the microorganisms and thus structure, otherwise which is invisible to the naked eye. The magnifications attainable by microscope ranges from x100 to x4 lakh. And the Antonin van Leeuwenhoek was the first who discovered the microscope and was the first to report his observations with accurate description and drawings. During his lifetime, he made more than 215 microscopes consisting of home ground lenses mounted in brass and silver. Next, the microscopes are two categories, that is light microscopy and the electron microscopy, which is depending upon the principles on which the magnification is based. The first type of microscopy is the light microscopy. In the light microscopy, the magnification is obtained by a system of optical lenses using the light waves. And the light microscopy further divided into four different microscopies, that is bright field microscopy, dark field microscopy, fluorescent microscopy, and phase contrast microscopy. So the first type of microscopy is the bright field microscopy. In bright field microscopy, the microscopic field is brightly lightened and the microorganisms appear dark because they absorb some of the light. Microorganisms do not absorb much light, but staining them with a dye greatly increasing their light absorbing ability, resulting in the greater contrast and color differentiation. Generally, microscopes of this type produce a useful magnification of about x 1000 to x 2000. At magnification greater than x 2000, the image becomes fuzzy. Then, what is resolving power? The basic limitation of the bright field microscope is 1 knot of magnification, but of resolving power. The ability to distinguish two adjacent points as distinct and separate. The greater magnification and the greater resolution is not beneficial. The resolving power of a microscope is the function of the wavelength of light used and the numerical aperture of the lens system. The numerical aperture means the sign value is multiplied by the refractive index n of the medium filling the space between the front lens and the cover slip. That is Na equal to n sine theta. The maximum numerical aperture of a dry objective is less than 1 and the oil immersion objective has an numerical aperture value of slightly greater, that is 1.2 to 1.4. The wavelength of the light used in optical microscope is also limited. The invisible light ranges between 400 nanometer to 700 nanometer. So the resolving power of the optical microscope is restricted by the limiting values of the numerical aperture and the wavelength of visible light. Here we can see an bright field microscope. Next type is the dark field microscope. The effect produced by the dark field technique is that of a dark background against which objects are brightly illuminated. This is accomplished by equipping the light microscope with a special kind of condenser that transmit a hollow cone of light from the source of illumination. Most of the light directed through the condenser does not enter the objective. The field is essentially dark. The diffracted light will enter the objective and reach the eye. Thus, the object or microbial cells will appear bright in a dark microscopic field. This is an image of the dark field microscope. Here we can see the condenser will um, transmit the hollow cone of light from the source of illumination. Next type of microscope is the fluorescence microscopy. Many chemical substances absorb light after absorbing the light of a particular wavelength and energy. Some substances will then emit the light of a longer wavelength and lesser energy content. Such substances are called the fluorescent and the phenomena is termed as the fluorescence. Application of this phenomena is the basis of fluorescence microscopy. The microorganisms are stained with the fluorescent dye and then illuminated with the blue light. The blue light is absorbed and the green light is emitted by the dye. In this fluorescent microscopy, there are two different types of filters are there, that is exciter filter and the barrier filter. The function of the exciter filter is to remove all not blue light. Barrier filter block out the blue light and allow the green light to pass through and reach the eye. The barrier filter are selected on the basis of the dye used. This is an image of the fluorescent microscopy. And the next and last of the light microscope is the phase contrast microscopy. It is extremely valuable for studying living unstained cells and is widely used in the applied and theoretical biological studies. It uses a conventional light microscope fitted with a phase contrast objective and phase contrast condenser. This special optical system make it possible to distinguish unstained structures within a cell which differ only slightly in their refractive indexes or thickness. The principle of phase contrast microscopy is based on the fact that light passing through one material and into another material of a slightly different refractive index or thickness will undergo a change in phase. These differences in phase or wavelength front irregularities are translated into variations in the brightness of the structures and hence are detectable by the eye. With phase contrast microscope, it is possible to reveal the differences in cell and their structure. This is an image of the phase contrast microscope. And the next type of microscope is the electron microscopy. As the name suggested, uses a beam of electrons in the place of light waves to produce the image. Specimens can be examined by either transmission or scanning electron microscopy. The first type of electron microscope is the term, that is the transmission electron microscope. Electron microscope provide tremendous useful magnifications. The electron microscope uses electron beams and magnetic field to produce the image. With an electron microscope employing 16 to 18 kilovolt electrons, the wavelength is only 0.05 angstrom. The resolving power of the electron microscope is more than 100 times that of the light microscope, and it produces useful magnification up to x4 lakh. For electron microscopy, the specimen to be examined is prepared as an extremely thin dry film on small screens, and it is introduced into the instrument at a point between the magnetic condenser and the magnetic objective. Numerous techniques are available for use with electron microscope, including the shadow casting, negative staining, ultra thin sectioning, etc. This is an image of the TEM. The next type of electron microscope is the scanning electron microscope or SEM. In scanning electron microscope, the specimen is subjected to a narrow electron beam which rapidly moves over the surface of the specimen. This causes the release of secondary electrons and other types of radiation from the specimen surface. The secondary electrons are collected by a detector which generate an electronic signal. These signals are then scanned in the manner of a television system to produce an image on a cathode ray tube. The scanning electron microscope lack the resolving power obtainable with the transmission electron microscope, but has the revealing a striking 3D structure. But it having some limitations that are the specimen being examined is in a chamber under very high vacuum. Thus, the cell cannot be examined in living state. The drying process may alter some morphological characteristics of the cell. Thus, the technique have low penetration power of the electron beam. And this is an image of the SEM. And thank you for listening me.